go. Okay, so we said that DNA was the instructions for making protein, right? But we need a little bit of information on how this happens. Now, raise your hand if you have any freshman year. Okay, you might remember this little activity. We have to do it again because those who had Miss Schroeder never did this activity.
Sounds about right.
Okay, I need Jonathan, I need you to be a good sport for me, okay? And Jonathan's going to be our ribosome. What's the job of the ribosome? Make the protein. So what we're going to do is I'm going to be the nucleus, and I have the DNA. And what's the DNA? Instructions for making the protein, right? So what we're going to do is we're going to pretend like this class is the nucleus, and you're going to be the ribosome. So I want you to go out to the hallway, and you're going to use these instructions to build the protein. Okay, so go out.
We're the nucleus.
We're in the nucleus. You're going to go to the cytoplasm where the ribosomes are. Okay, so Jonathan is our ribosome. Okay, once you get out there, I want you to go ahead and start building the protein. Go on. All right. You ready? Does anybody see a problem?
He doesn't have the instructions.
He doesn't have the instructions. I have the instructions. I don't know what he's doing out there, but it's sure not using these instructions to build the protein.
How long he'll wait out there before finally realize what they have?
Let's see if he knows what the problem is.
He's still in.
Senor, did you build that protein? I have the instructions.
Couldn't do it without
Don't have 15 instructions.
You can't have this copy.
We'll just, there's a copy machine downstairs.
Okay, so he said, can I just take the instructions? Let me ask you a question. Jonathan, how many copies of the DNA does any one cell have? One. And how many ribosomes would you suppose, roughly, that any one cell probably has?
Let's see.
One big rush.
Have you ever seen a picture of a cell and all the little dots that were labeled ribosomes? Right? Lots of them. So, number one, it would not be very efficient of us to have one DNA and send it to a ribosome, right? Number two, you didn't know this, but I'm going to tell you now, the double helix is too large to fit through a nuclear pore. Okay? So, it can't leave the nucleus. That's a problem. You need some instructions. Okay? Number three. This DNA is the one, Kayla, it's the one and only copy that this cell has. And if something happens to it, death will surely follow. Okay?
Sound like Miss Santiago when she threatened my life.
So this DNA,
Someone is coming for you, and death will surely follow.
I tell this to my freshmen, and some of you probably heard the story, but
You teach freshmen too?
no, when I taught freshmen.
Does it remind
Yes. Imagine you're walking to school, and on your way to school, you find a
$1.
no $10 million diamond. I can see you guys don't remember the story. $10 million diamond, okay? And you get this $10 million diamond, and you're so excited, so you get it, and you run all the way home. You don't even go to school that day. You just run straight home, and you call your best grandmother in Tennessee, and you say to her,
Wait, my best friend?
"You're the only 10 I see." No, just kidding. Then you say to her, "Grandma, you're not going to believe what I found. This $10 million diamond is so awesome. It's going to change my life forever." And your grandmother, she's so sweet. She says, "Oh, honey, that sounds wonderful. Why don't you send it to me so I can see it?" What do you mean, no? You're not going to send your grandmother a $10 million diamond? Don't you trust her?
Grandma's a backstabber.
So, this $10 million diamond is analogous to the cell's what? DNA. What will you probably send your grandmother? A picture. So when Jonathan is out there in the hallway and he says, I need some instructions, I don't know what I'm doing, I'm not going to send him my one and only copy of DNA that I have in the cell, because to me it's like a $10 million diamond. Number one, if I could even fit it through the nuclear pore, it might get damaged and I would die. So I need to make a copy. I need to take a picture. I need to send something else, a replica of some sort, right? I want to send RNA. I want to send RNA. So what we need to know today is, how do we make that copy? There's two parts to building a protein. Copying the DNA for that purpose, and then actually building the protein at the ribosome. That's two steps. We are only learning the first step today, because I have too much to tell you about the first step. So, we already know genes code for proteins, blah, blah, blah. This is called the central dogma of biology. You got the flow of information. If we do replication, we make more DNA. If we do transcription, we make RNA. By the way, you might have heard of something called tRNA, mRNA, rRNA, siRNA, IBRNA. That last one. See what I did there? I made it up. Okay. Transcription makes all kinds of RNA, not just mRNA. I need you guys to realize that. Okay, so transcription, usually we talk about it in terms of mRNA because our goal is to build a protein, but transcription can make all kinds of RNA. And then we do another process, the second step, which is called translation, in order to build a protein. Where does transcription occur? In the nucleus, because that's where my DNA is, right? Where does translation occur? At the ribosome. Okay. So, before we can talk about how to make RNA, you need to know what RNA is and what it is not. It is a single-stranded nucleic acid. It has ribose instead of deoxyribose. Yeah, Melanie? High five. Okay. And it has 4 nitrogen bases. There is one difference. Uracil instead of thymine. Guys, if I show you a DNA sequence or a nucleotide sequence, and I ask you if it's DNA or RNA, the one major indicator is the presence of U or a T. If I'm presenting you with a U, you know it's RNA. There will never be a T in RNA. Let me state that again. There will never be a T in RNA. And I'm stating that because I ask kids about tRNA, and they think that it has a T in it because of the name tRNA.
Silly.
It's not true. There's no T's in RNA, just in the name tRNA, but not any bases of T's. Got it? Okay, ever. So U will pair with A, and C will pair with G. We say it's single-stranded, not double-stranded. Being single-stranded allows it to fit through nuclear pores. Wow, you guys are on it. Here's some different kinds of RNA here. mRNA, tRNA, RNA, siRNA. There was an AP exam question 5 years ago, and it was explain the different kinds of RNAs and their functions and how they're used, something like that. Okay, so here's this slide. Messenger, because it's taking the message from the nucleus to the ribosome. So it turns out that transcription, the actual process of building the RNA, only needs one slide because it's that simple. Here's how it works. RNA polymerase binds to a DNA strand at what location? The promoter region. Now, in eukaryotes, the promoter region has a ta-ta box, a sequence that says TA, and that's the promoter region. That, my friends, is where RNA polymerase will bind. Now, RNA polymerase is a very versatile enzyme because it can unzip and copy at the same time. No need for a helicase, No need for ligase, no need for DNA polymerase 3 or 1. It's awesome. RNA polymerase unwinds the DNA. Well, first it binds to the promoter region, right? Then it unwinds the DNA, and it will add nucleotides to a growing strand. What kind of nucleotides are we talking about? RNA nucleotides. And what direction can we build our new RNA strand? Well, same direction we build our DNA strand, 5' to 3'. It's easy because it's the same. See this green strand here? This is my mRNA that I'm making. It's reading the blue strand. The blue strand that it's reading is called your template strand. Your coding strand is the strand that it doesn't read, up here. Can somebody explain to me why the coding strand is the one that's not read? Yes. Yes. If I match the bottom, then the new mRNA turns out identical to the coding strand, right? The only difference is U instead of T, but all the bases will be in the same place. So we say this is our template, and the one not being copied is the coding strand because they turn out identical. Questions on this part? It's pretty easy. So let's recap. RNA polymerase, which is an enzyme, can bind to the promoter region. Once it's attached, it will build an RNA molecule. It reads the DNA in a 3' to 5' direction, but it builds the new RNA in 5 to 3. As soon as it passes a sequence of DNA, that DNA is re-zips. Where does RNA polymerase bind? Promoter region. And the promoter region has a Tata box, right? We can also, if we're talking about gene regulation, we can also have what here between the promoter and the gene? An operator. And the operator can have a regulator or a repressor. So a promoter, Tata box, maybe an operator depending on if we're regulating that gene, repressor, and then the gene. There was one other thing I think I forgot to mention earlier. Oh no, I did mention. Remember I told you there was something else required for this to happen, but I'll tell you later. I said this can bind. It needs transcription factors. These are proteins that are required for RNA polymerase to bind to the promoter region. They're called transcription factors. Can you repeat that? Transcription factors are proteins that are required for RNA polymerase to bind and for transcription to begin. They're called transcription factors. Here in this picture, they're represented with just a bubble. Proteins required for RNA polymerase to bind and for transcription to begin. I can show you guys like an animation, and you'll see like all of these weird shaped molecules coming to the site, and you'll be like, what are all those things? Guess what they are? Transcription factors. We have lots of them. Like maybe up to 10 required for one polymerase to transcribe. If I write, let's say that everything worked out, RNA polymerase came, binded to the promoter region, the transcription factors were there, transcription began, RNA polymerase went down the line, transcribed the whole gene. And now we have this RNA. The RNA that's produced from transcription is called an RNA transcript. The RNA produced from transcription is called an RNA transcript. It's not ready to leave. It has to be processed. It has to undergo post-transcriptional processing, where we cut out some DNA that's not needed, package it properly. So let's take a look at that. In any given gene, now this is eukaryotes, okay? In any given gene, we have sections of DNA that don't code for anything. This is really bizarre. Guys, I'm telling you right now, in the gene that codes for your cardiac muscle, there is DNA that doesn't code for anything spread out in that gene. We have some theories on what that DNA is, like why it's there, but we don't really know. So when we transcribe a gene, we get this RNA transcript that has all this extra stuff in it that we don't want. We got to take it out. So what we do is we splice this. We literally have something called a spliceosome. I know it sounds like I made that up, but it's real. Okay, spliceosomes that cut out the introns. DNA that doesn't code for anything is called an intron if it's in a gene. So what I say is cut out the N and keep the X. If you can remember that, you'll keep them straight. Cut out the N and keep the X. Cut out the introns. We're going to keep the exons. Exons are the actual coding DNA. What is this mRNA called when it's before it's processed? RNA transcript. What is the DNA called that doesn't code for anything inside of a gene? Good. And what's the enzyme that will cut the introns out? Very good. Spliceosome. Just like it sounds. Actually, I think it's on your notes too. So take a look at this picture. This is pretty cool. If you guys can see here, we cut out the introns, right? Do you see that my gene now has sections where it was spliced together? See where it has sections that was spliced together? You see that? You guys see? What if I combine these sections in different ways? I will create a variety of proteins from one gene if I combine them in different ways. So actually, remember what I told you? That you have 21,000 genes? And I asked you if you could make 21,000 proteins? And you guys said no. And I said, actually, you can make more than that? This is how. So you have a gene, and we can combine these sequences in different ways to make a variety of proteins from that one gene. I don't have a real example for this, but I think you could find one if you want. It's still not ready for export. Meg, do you know what it needs before it can be exported? Remember this? The RNA needs a 5' GTP cap. And now we are experts in biochemistry. We know what GTP is, correct? It's an energy molecule. Guanine triphosphate, a nucleoside, and it can be used for energy. So we're sending this little RNA out with a little package of energy to help it do what it needs to do when it gets where it's going. On the other side, we have a 3' poly A tail. This is just a really long sequence of adenines that help, in case this gene gets broken or damaged or something on the end, it won't damage the important part. It's like a buffer. It's like a package. It's like peanuts. Packing peanuts.